So, in this short video, you will learn more about the Campylobacter species. Campylobacter species are small, curved, motile, gram-negative bacilli, and most of them are pathogenic and associated with a wide variety of diseases in humans and other animals. They are microaerobic inhabitants of the gastrointestinal tracts of various animals, including poultry, dogs, cats, sheep, and cattle, as well as the reproductive organ of several other animals. Campylobacter species produce three syndromes in humans, namely the febrile systemic disease, periodontal disease, and most commonly gastroenteritis. Campylobacter species are the causative agents of gastrointestinal or extraintestinal infections. An increase in extraintestinal disease, including meningitis, endocarditis, and septic arthritis, has been reported in patients with acquired immunodeficiency syndrome, or otherwise known as AIDS, and other immunocompromised individuals. Within the genus Campylobacter, Campylobacter jejuni and Campylobacter coli are commonly associated with infections in humans and are transmitted via contaminated food, milk, or water. But remember that Campylobacter does not multiply in food.
Campylobacter is a common bacterial cause of illness in humans. It grows in the intestines of birds and mammals. Symptoms include bloody diarrhea along with nausea, vomiting, abdominal pain, headache, and muscle pain. Symptoms usually dissipate after 1 week, though in severe cases, long-term consequences such as Guillain-Barré syndrome may occur. If you experience any of the symptoms, seek medical care and ask your healthcare provider to take a stool sample. Also, document what you've eaten in the past 72 hours.
So hi everyone, today let's talk about Archobacter. So what are Archobacter? So Archobacter is a genus of gram-negative spiral-shaped bacteria in the Epsilon Proteobacteria class. It shows an unusually wide range of habitats, and some species can be human and animal pathogens. Species of the genus Archobacter are found in both animal and environmental sources, making it unique among the Epsilon Proteobacteria. So this genus currently consists of 5 species: the Archobacter butzleri, Archobacter cryophyllus, Archobacter skirrowi, Archobacter nitrophidilis, and Archobacter sulfidicus. So Archobacter has emerged as an important food-borne zoonotic pathogen, causing sometimes serious infections in humans and animals. So as you can see in my background, that's how Archobacter looks like. It looks like a worm. Lots of worms. Archobacters are primarily transmitted through contaminated food and water sources. These organisms have been reported from chickens, reptiles, meat, and vegetables, and from humans in different countries. There you can see the pathogenesis and transmission of Archobacter. So Archobacter, after entry through the digestive tract, colonize the intestine. Several mechanisms like production of cytotoxin affecting expression of tight junction proteins, release of pro-inflammatory cytokines, and epithelial barrier dysfunction lead to cell death or apoptosis. So the organism is released into the environment where it contaminates water and food sources, thereby entering the human food chain. So that is how the Archobacter species transmitted. So Archobacters are implicated as causative agents of diarrhea, mastitis, and abortion in animals, while causing bacteremia, endocarditis, peritonitis, gastroenteritis, and diarrhea in humans. How can we prevent acquiring or getting the Archobacter infection? So, proper preparation and cooking of all foods derived from animal sources, particularly poultry, will decrease the risk of transmission. That would be all for the Arcobacter. Thank you. Bye.
Good day, everyone. This is Nicole Marapao. I will be discussing the Helicobacter species. General characteristics. In 1983, spiral-shaped organisms resembling Campylobacter species were isolated from the human stomach. These organisms were named Campylobacter pylori. The genus Helicobacter was established in 1989. C. pylori was renamed Helicobacter pylori. Approximately 32 species are included in this genus. The genus Helicobacter consists of curved, helical or spiral, or fusiform microaerophilic gram-negative rods with or without periplasmic fibers, with the majority of species exhibiting urease activity. The organisms may appear coccoid or spheroidal if cultivated for long periods. Human isolates include human pathogens discussed here include epidemiology and pathogenesis. Helicobacter pylori's primary habitat is the human gastric mucosa. The exact mode of transmission is still unknown. An oral to oral, fecal to oral, and a common environment source have been proposed as possible routes of transmission. Research studies suggest that mother to child transmission is the most probable cause of intrafamilial spread. In this slide, we can see the following habitats of the Helicobacter Helicobacter species. Helicobacter pylori is capable of colonizing the mucous layer of the antrum and fundus of the stomach but fails to invade the epithelium. Motility. It allows the Helicobacter pylori to escape the acidity of the stomach and burrow through and colonize the gastric mucosa in close association with the epithelium. Helicobacter pylori is non-invasive. Untreated colonization persists despite the host immune response. Spectrum of disease. Helicobacter pylori causes gastritis, peptic ulcer disease, and gastric cancer. Infection with this organism is also a risk factor for the development of atrophilic gastritis, gastric ulcer disease, gastric adenocarcinomas, and gastric mucosa associated lymphoid tissue or MALT lymphomas. Enterohepatic helicobacters. This inhabits the intestinal and hepatobiliary tracts of various mammals and birds. Species that are capable of causing diseases in humans include the following. Helicobacter phenoliae have been isolated in cases of proctocolitis, gastroenteritis, neonatal meningitis, skin rashes, and bacteremia in immunocompromised patients. Laboratory diagnosis. Specimen collection, transport, and processing. There are no special requirements for the collection, transport, or processing of stool or blood specimens for Helicobacter species. Blood samples for serologic diagnosis of Helicobacter species infection may be collected, transported, and processed by standard methods. Additional samples may include urine for antigen testing and saliva samples for serologic tests. These specimens do not require any special processing or treatment. Direct detection. Pathologists use the Warthin stari or other silver stains and gemsa stains to examine biopsy specimens. However, because of the presence of bacterial atypical morphologies, the results may not be interpreted correctly. Sampling error may occur during processing, resulting in no identification of the organisms. Cultivation. Stool specimens submitted for culture of Helicobacter cyneidae and Helicobacter phenoliae are inoculated onto selective media used for Campylobacter isolation but without cephalothin, such as Campy-CVA. The recovery of H. pylori from tissue biopsy specimens include gastric antral biopsies, non-selective agar media, including brain heart infusion agar, chocolate agar, and brucella agar with 5% sheep blood. Wilkins Shalgren agar and trypticase soy agar have resulted in successful recovery of the organisms. Selective agar such as Skiro and modified thiamartin agar also support growth. Recently, the combination of a selective agar and a non-selective agar was reported as the optimal combination for recovering H. pylori from antral biopsies. Approach to identification. Colonies of Helicobacter species may require 4 to 7 days of incubation before small, translucent, circular colonies are observed. Culture plates should be reviewed daily for a minimum of 10 days before a negative culture is reported. Organisms are identified presumptively as H. pylori by the typical cellular morphology and positive results for oxidase, catalase, and rapid urase tests. H. pylori, H. cyneidae, and H. phenoliae are definitely identified by using a similar approach to Campylobacter species. Antimicrobial susceptibility testing and therapy. Except for metronidazole and clarithromycin, most laboratory susceptibility assays are unsuccessful in predicting clinical outcome. Routine testing of H. pylori isolates susceptibility to metronidazole is recommended using the e-test and agar or broth dilution methods. Therapy for H. pylori infection is problematic. Current regimens Current regimens recommend triple drug therapy, including a proton pump inhibitor, clarithromycin, and either amoxicillin or metronidazole. An alternative and simple regimen for patients with metronidazole resistant strains includes omeprazole or lansoprazole and amoxicillin or clarithromycin. Helicobacter species associated with enteritis and proctitis may respond to quinolones. However, appropriate therapy has not been established. Lastly, prevention. There are no vaccines available for Helicobacter pylori. However, several vaccines are under development.